In this video, we're going to talk about some of the ways that transgenic animals are used for genetics research. First, let's go over the learning objectives for this video. By the end of the video, you should be able to explain what a transgenic organism is, explain how transgenic organisms are generated, and discuss some examples of how transgenic organisms are used. Let's start by reviewing the definition of a transgenic animal. A transgenic animal is an animal that contains foreign DNA in its genome, either in the form of an insertion or a substitution. And while we're focusing here on transgenic animals, it is also possible, of course, to make other types of transgenic organisms, like transgenic plants or transgenic bacteria. So let's first go over some of the common uses of transgenic organisms, and then we'll discuss two different examples in more detail. One reason you might make a transgenic animal is to figure out which cells in the body express a gene of interest. In this case, you might make a transgenic animal containing a DNA construct in which an enhancer or promoter for the gene of interest drives expression of GFP. In these animals, wherever the enhancer or promoter element would normally drive expression of your gene of interest, you'll see expression of GFP. For example, let's say you do this with C. elegans and you see something like this. In this case, you can see expression of GFP in head neurons. And so then you know that your gene of interest is expressed in head neurons. Now, this approach works in simpler organisms like C. elegans, where the regulatory elements that control gene expression are less complex. And even in C. elegans, you do need to use other approaches to confirm gene expression patterns. But this approach of using transgenics can give you a general idea of where in the body your gene of interest is expressed. Another reason to make a transgenic is to fluorescently label particular cells or cell types in the body. For example, let's say you want to make a mouse with green glowing skin. You can make a mouse containing a DNA construct with a skin cell promoter driving expression of GFP. So here you can see some transgenic mice with green glowing skin next to some wild-type mice. And you can buy these mice in pet stores now along with green glowing fish. And then finally, another use of a transgenic is to make an animal that overexpresses or misexpresses a normal or mutant gene. To do this, you would make an animal containing a DNA construct in which your gene of interest is expressed under the control of a promoter that drives expression in the cells or tissues of interest. So now we're going to take a look at two different examples of this use of transgenics, where you're trying to overexpress a particular gene of interest. The first example we're going to talk about is the transgenic mouse that is sometimes called the super mouse. To give you the relevant background information, there's an enzyme called phosphoenolpyruvate carboxykinase, or PEPCKC, that plays a role in glucose metabolism in the kidney and liver. However, this PEPCKC gene is also expressed elsewhere, including in skeletal muscle. But it wasn't clear what the function of this enzyme is in these other places in the body, like the skeletal muscle. So the research question here was, what is the function of this PEPCKC gene in skeletal muscle? To address this question, researchers made a transgenic mouse that overexpresses the PEPCKC enzyme in skeletal muscle. And then the researchers conducted a number of different behavioral tests with this transgenic mouse to see if its muscle function was different from that of a normal mouse. To make the transgenic mouse, the first step is to generate a DNA construct for overexpression of the PEPCKC gene in skeletal muscle. And this involves subcloning, where you PCR amplify different fragments of DNA from mouse genomic DNA, and then you ligate them together. So first you PCR amplify the promoter for an actin gene that is normally expressed in skeletal muscle. And then you PCR amplify the PEPCKC gene. And usually when you do this, you actually generate PEPCKC cDNA. So basically you have the gene sequence without introns. And you want to do this because introns often contain regulatory elements, and these regulatory elements might cause your gene to be expressed in places other than the skeletal muscle. Okay, so then you ligate the promoter for the actin gene in front of the PEPCKC gene. And then the next step is to make a mouse that expresses this transgene by injecting the transgene into a mouse embryo. And then in the transgenic mice that result, the PEP-CKC gene will be overexpressed in skeletal muscle. So how do you actually go about making the transgenic mouse? First, you inject the DNA transgene that you made into a fertilized ovum. In this case, as I mentioned, the transgene has the actin promoter driving expression of the PEP-CKC gene. And at some frequency, the transgene will integrate into one of the mouse's chromosomes, although you don't know ahead of time where the transgene will integrate. An important thing to note is that when you make a transgenic, you typically get many copies of the transgene integrating into the gene all together as a large concatemerized array. So genes that are present on transgenes are often expressed at higher levels than they would normally be expressed endogenously. Okay, you inject the transgene right after the ovum has been fertilized, before the pronucleus from the egg and the pronucleus from the sperm have fused. And in particular, you usually inject the transgene into the male pronucleus, since the male pronucleus is larger than the female pronucleus. You then implant this ovum into a pseudopregnant female. And then some of the progeny from this mouse will be transgenics. And you can identify the transgenics by testing their tail DNA for the presence of the transgene. Then finally, the last step is to breed the mice to homozygosity. And basically, this means that you mate transgenic mice together, and then you select progeny that are homozygous for the transgene. For example, if your transgene happened to integrate on chromosome 3, then you want transgenics that have the transgene on both of their third chromosomes. Once you have mice that are homozygous for the transgene, you then test them to study their phenotype. So in this case, the transgenic mice we're talking about have this DNA construct integrated in their genome. And as a result, they overexpress this PEP-CKC gene in skeletal muscle. These mice were then subjected to a number of behavioral tests to assess their muscle function. And the goal here was to see whether overexpressing the PEP-CKC gene in muscle alters muscle function. These behavioral tests revealed that the transgenic mice actually have greatly enhanced muscle function, and as a result, they were extremely active relative to normal mice. So in this figure, you're looking at 4 different measurements of activity, and in each panel, the gray bar shows the wild-type response, or the response of the wild-type mice, and the black bar shows the response of the PEP-CKC overexpression transgenic mice. And in each case, you can see that the transgenic mice are much more active than the wild-type mice. So for example, the top left graph shows the total distance traveled by the mice in a 22-hour period. And you can see that the transgenic mice cover much more distance than the wild-type mice. And the top right panel shows the average speed of the mice, and you can see that these transgenic mice move much faster than wild-type mice. So basically, all of these different measurements indicate that the transgenic mice are extremely strong and active, and this is why these mice were nicknamed super mice. You can see this perhaps even more clearly in this video, which was made by the authors of this paper. In this video, the transgenic super mouse is in the front, and the wild-type mouse is in the back. And both of the mice are running on a treadmill. And basically what you'll see is that the transgenic super mouse keeps running even long after the wild-type mouse has given up and fallen off the treadmill. So let's watch the video. So what was learned from making these transgenic mice that overexpress the PEP-CKC gene in muscle? Well, first, we learned that the transgenic mice have more efficient metabolism in muscle cells. Second, the transgenic mice were actually found to be healthier and longer lived than wild-type mice. Overall, these results indicate that the PEP-CKC gene is a key regulator of metabolism in muscle cells. And these results raise the intriguing possibility that enhancing PEP-CKC function in human muscle tissue might also be beneficial for human health. Now we're going to talk about a completely different example of the use of transgenics that has to do with the food industry, and specifically with the production of transgenic Atlantic salmon. Atlantic salmon are grown in a lot of fish farms for human consumption. And the reason for trying to make transgenic salmon is that Atlantic salmon grow very slowly, and they don't reach their full size for at least 2 years. In addition, they're pretty small even when they reach their full size, as you can see from the picture. So the research question was, is it possible to engineer transgenic salmon that grow faster and that end up larger? And if so, this could make Atlantic salmon easier to farm, which would make them more available in fish markets. The approach researchers took is to try to make a transgenic fish that overexpresses a growth hormone gene, in the hope that the fish would grow larger and reach their full size sooner. To make a transgenic salmon that overexpresses the growth hormone gene, the first step is to generate the DNA construct for overexpression of a growth hormone gene. And the goal was to generate a construct that overexpresses a growth hormone gene throughout the body of the salmon. In this case, researchers used the promoter for an ocean pout antifreeze protein gene because this promoter drives expression throughout the whole body of the salmon. And they used this promoter to drive expression of a salmon growth hormone gene. So using standard DNA subcloning techniques, they generated a DNA construct containing the promoter for the antifreeze protein gene upstream of the growth hormone gene. In salmon that express this construct, the antifreeze protein gene promoter will drive expression of the growth hormone gene throughout the body of the salmon. Once you have this DNA construct, the next step is to inject it into salmon eggs. So let's briefly take a look at how you do this. First, you take fertilized salmon eggs. Then you just inject the DNA construct into the fertilized eggs. And then you let the fertilized eggs grow up into fish, and a subset of these fish will have incorporated the transgene into their genome and will be transgenics. What researchers found from doing this is that the transgenic salmon grew to be much larger than their wild-type counterparts. You can see that in this photo where the yellow arrow indicates a transgenic fish side by side with a wild-type fish. And as you can clearly see, the transgenic fish is much larger than the wild-type fish. In addition, as you can see from this graph, the transgenic salmon, which are called aqua-advantage salmon, not only end up larger, but they also grow faster than wild-type salmon. What this graph shows is their weight as a function of their age. And you can see that the transgenic salmon are larger from an earlier age, and they reach their full size sooner. So overexpression of growth hormone does in fact result in larger fish that grow faster. These aqua-advantage transgenic salmon are currently being marketed in Canada for food, although they're not yet approved for sale in the United States. To conclude, I hope from these examples you have a better understanding of what transgenic organisms are and how they're generated. You should also be able to discuss how transgenic organisms are used using the PEP-CKC transgenic mice and aqua-advantage transgenic salmon as examples. And finally, I want to mention that there are obviously many scientific and ethical issues to think about when talking about the use of transgenic organisms in food and agriculture. And this is something I'd like you to continue to think about as we discuss transgenic organisms in class.